Hello, everybody. I'm SquishyMan, if you didn't know. I think my name is SquishyMan, and I think we're gonna be playing some sort of thing. We're gonna be playing Slaughterhouse, alright? Real bear. Romby Rubux, a member for 4 months. This guy's actually goated. W in chat to Romby Rubux. Rubux. Let's go. Romby Rubux. Okay. Right. There's this one thing. It's called Geometry Dash. This is the wrong one. Right, we're doing this. Let's no clip. What a noise right there. Alright, chat. If my name is Tim, YouTube isn't Chat, YouTube isn't working. Okay. Alright, we gotta do a goal. Let's do like 5 no, let's do like one $20 super chat. And I'll rename the stream to Steve. What a great idea. What the nuts? Hear me out! Atlantic Ocean.
Okay.
Ow! Why it be like that? Bro, it's always there, man. I'm gonna It's upsetting. Very upsetting. Chet. Who filled my kidney with fertilizer? Bro, it's literally just... Okay, that's good. That shows that we are not very stuck there. At like 8:30, I'm gonna just start streaming my biology homework. Cause I'm behind on it. And I have an exam coming, so I have to... Let's all learn about I don't know. Let's all learn about flipping biology together. Bad grades, man. No, grade of 67, man. 67. I'm so dead when I freaking unpause. Whoa. What? Sorry. Yep, oh, my bad. That's good, because at least we passed that one part that has the annoying stuff. Oh, what? Real bear.
It's my birthday 2 days in from Australia.
Yo! Ramos, what's up? Happy birthday from Australia, did I hear? And yours! The 2 blue W in chat. Happy birthday in 2 days. Okay, whoa. Happy birthday in 2 days. Oh, we're about to hit 12K as well. Oh, chat. Huh? What? Huh? Chat. Chat. W in chat for the 2. And yours with the 1. Everybody on YouTube, in the link in the description of the YouTube stream, you can go to Twitch and help us reach the amount of followers. Oh, we went up 4, chat. This is quite close. We are actually quite close. We are close. 12 people, come on. Lock in. Everybody on YouTube, click the Twitch link in the description and follow there too. Because we are trying to reach 12K. We are 12 away. Damn. Oh, one person did it. Come on. Chat, please. We're right there. We are right there. Oh. Ignore the ads. Ignore the ads, chat. I promise. We're right there. Come on. Sorry, what? For good luck. Yeah, the good luck actually pushed me pretty far. W in chat to friends. We are literally 8 away, bro. Please. Chat. We're right there. Chat. We are literally... Okay, hold up. I'll tell you what. That, okay, hold on. Does it let me scroll? Bro, I can't scroll. Oh, that's so annoying. Okay. I'm not human. I'm not human. Bro, we are literally 6 away. Bro, whatever. Social Blade. Sure, no, Social Blade's bad. Twitch Tracker. It's called Twitch Tracker. Come on. Streets. You know, I give up. We're using this. And we're 6 away. Come on. Yo, Steve with a weapon with the 10 Muxon. W, Steve in chat. This guy's actually goated. What is Muxon? Let's go to CAD here. 77 cents. In US, probably like, ooh, yeah, 55. W in chat. We are 6 away. Nope, let's not. I think I accidentally pressed it. Chat, we are, yes! 5 away. Now I am broke. No. Chat. Come on. We're right there. Ooh, 5. Everybody on YouTube, go to Twitch and follow. You're technically an OG if you sub before 12,000, and now's your last chance. So What else am I saying? Oh. So in the link of the description of the YouTube stream, you can go to Twitch. We're 3 away. 3 more spots. Who will take the spots? Who will be part of the mighty 12? Oh, no, chat. Somebody left it. 1 more spot open, I guess. Wah!
We reached our followers goal of 12,000. Thank you everyone.
Does that say we did it? I want to see it on here. I want to see it on here. Come on, we're literally 2 away. Okay. I'm resetting you, alright?
We reached our followers goal of 12,000. Thank you everyone.
No. I changed tabs for 1 second!
We reached our followers
Okay.
goal of 12,000.
W in chat. You know what? Fine, W in chat. Okay. Alright. You know, alright. Yeah. Okay. Great. Okay, I need to do biology, bro. Hold on, everybody. Alright, chat. Prepare to be bored. Holy, you guys are gonna be so bored. I love it. You guys want to learn about biology? We're gonna love this. Studying in chat, just, yeah, yap in chat. Meanwhile, I'll, like, I'll play you guys some music. Alright, what the? Bro, go down. Alright. Can I be the music to the ears of the chatters? Sure. Alrighty. You guys will not be able to see this.
Don't leak anything from your school.
Yeah, no, I'm only writing notes. I did a little studying beforehand, like, does terminology used narrow down anything? And no, really not. Because that's the only thing. The only factor could be the terminology I use, which rarely varies between, like, countries even, so.
Yeah, you should be good.
We should be good. Move you dumb Corsair. Even though I kinda need it. Okay, no, I'm gonna leave it here. I'll just be hiding screen. Also, while you're... Okay, hold up. Yeah, we'll just pop questions in there, and then I'm gonna do... Okay!
Wow.
Here we go.
See, this question is weird because it could go either way. The one on YouTube. In 2.1, I think VSU was way harder, but now I think Secupid Circles is way harder.
Yeah, that's weird. Is it because of physics, or?
Well, I like to call it the triple threat, which is the Corsair K70. So basically an 8,000 hertz polling rate device.
You didn't hear that? Yep.
CBF in 2.2.
You didn't hear that either. Yep.
CBF in 2.2.
I get you now. Alright. Where do I start? I kind of know what that is. These. You want, alright. Do we want to get into, okay. Hold on. What? Alright. So, right now we doing unit 1. What are we going to focus this stream?
Do lipids. I like the lipids. Yo, by the way, in America, on labels, I think we should change fats to lipids, because Americans look at fat on a nutrition label, and they think that automatically means bad. It doesn't.
Yeah, that is true. But, oh, we're learning about protein.
Of course they think. protein.
Of course. It's the one word that most people recognize, probably. I'm just studying for a bio exam.
I hated biology when I took it. I did so bad. I barely passed.
At least you got through it. Okay. So, made of elements carbon, hydrogen, oxygen, nitrogen. Sulfur is present too. That's crazy. If you can read that, I am impressed. If you can even see it on this camera, bro. Holy. Yeah, holy. I got the greatest handwriting. Sulfur is often...
I don't think it's AP Bio. Do they have AP classes in Canada?
Yes.
Okay.
And it's the highest it can go. What this is.
Yeah, same here.
Of. Well, yes, as we know, proteins are just a bunch of amino acids. Oh, dude, that's a nostalgia from last bio where you learn about like protein synthesis. Do you guys call it something else? Or do you guys also call it protein synthesis?
Probably. I don't remember as well now.
Doesn't ring a bell.
Well, bear.
Fake Pural resubbed 3
Yeah,
times.
give you some
Wowie
notes.
mama.
Wowie mama.
Wowie mama. Oh, you're sending me
Biology
notes.
involves the study of life from the smallest molecules to entire ecosystems.
Yep. Well, it's human
Explains
biology we're
how
doing.
cells function,
Oh,
how
human
energy moves
biology.
through living systems,
Sorry.
and how DNA controls traits through processes like replication, transcription, and translation.
That's
Core ideas
part
include
of...
cellular respiration,
Wait, are you talking about
photosynthesis, homeostasis, enzymes,
homeostasis?
and the structure of
What is
important
homeostasis?
molecules such as proteins, lipids,
I don't know.
and
That's
nucleic
ChatGPT
acids.
generated, bro. I'm not that
covers
smart.
inheritance patterns, mutations, gene expression.
Okay, I don't know how to focus. At least I know all the terms I mentioned, so we're chilling. Okay. So yeah, they're basically... Proteins are a bunch of amino acids. Amino acids have 2 different parts here. Let's zoom in here. Then what can I like chat, we're all learning now. Can I flip this? Does OBS do that? How do you rotate in OBS?
Oh, there's a way to do it. Let me open it up real quick. I'll help you. You gotta go to like cropping settings, I think. Let's see. Let me go to my camera scene. It's gotta boot up. What the hell did Bennett GS just send in the Twitch
I have
chat?
no idea.
That's getting deleted. Okay, I won't do
Holy.
600 seconds. I'll get rid of the 600 seconds, but yeah.
What is that thing we do?
Okay, right click the camera scene, the full like literally just right click your preview and then hover over transform and like go to the arrow.
Slow down. Go over the scene.
Literally, yeah, like literally like the preview you get that you look at on your Streamlabs or whatever you use.
Oh yeah.
Right click that.
Transform.
Transform, then rotate 180 degrees.
Hey, look at that. Hey. Hey, now you guys can see how bad my handwriting is. Even better. All right. So basically, Oh yeah, classic. Or not classic, but any GD player has bad handwriting. Okay, so in an amino acid, and this is what proteins are made of. Usually there's a bunch of amino acids. Pretend these are amino acids. There's like a bunch of them. Let's get that alpha helix, all of that in there. They're all connected via polypeptide bonds. Poly meaning many, peptide meaning shit. I need to study that. What is a peptide? No, isn't a peptide just another word for? Hold on. This is biology. It's just remembering. I think a peptide is just a single amino acid. So amino acid, or just peptide. But pretty much, I think it does dehydration synthesis. Yeah. So here's what happens. So pretty much, let's draw let's draw some here. Let's draw like a glycine. H. So hydrogen attached to a nitrogen attached to a hydrogen attached to a carbon. This carbon is called a radical group. So pretty much there's a carbon attached to a hydrogen up here, and then there's a radical group. This is pretty much the radical group is what distincts the amino acid from the rest. This is what distincts it. All right. Okay, hold on. I gotta make this a title. Proteins. But otherwise, it's pretty much all the same. There's the, what is this? The amine group. Hold on. The amine group or something. Hold on. Stick with me now. The amine. It was weird. Hold on. Stick with amine group. Yeah. And then there's, isn't it like a carboxyl? Bro, carboxy terminus. Sure. This third is the amine group. And then there is the radical group. So this doesn't change. And then there is this is pretty okay. Actually, I'm gonna talk about it. Then you got a carbon with an OH. Shoot, that's yeah. Equals so it's calling it a carboxy terminus here, but I've heard another word for it, so I'm not really, I don't like that. There's another word for it. I swear there's another word for it. Hold on with me now. Carboxylic acid. No, I'm confused. Wait. I gotta find it. What is it called? Hold on. What are they called? Help. Chat. I forgot what they're called. Oh no. Dude, this is all biology is. It's just remembering all these flippin' things. Alright. You know what? We'll figure it out. This and this. We're just gonna call it the carboxy terminus. Alright. Close enough. Oh yeah, this also... No, that's right. That's right too. Oh, so this is a double bond to an oxygen. Oh, weird. Okay. That is actually... That would have a neutral pH, wouldn't it then? Huh. I didn't even take chemistry, so I'm kind of learning 2 courses at once here. Anyways, so the reason that it starts with an amine group and ends with a carboxy, what again? Carboxy terminus. All right, we're barely getting through. And this is like a glycine pretty much. What happens is there's dehydration synthesis. So this is exactly what you'd expect. Dehydration, so getting rid of water, and then synthesis, so it goes together. It together, all right? So yeah, it forms water and it goes together. We're pretty much, you see there's an OH and an O here. And then you see how there's an H and an H here. Pretty much these 2 H's attached to this O, which is 2 H's, that's an O, that's an H2O. So off that goes, and then you're stuck with an OH plus an N. So then that synthesizes, and then that's a dipeptide. So now you have 2 peptides formed by 1 peptide bond. There you go. That's dehydration synthesis. The opposite should be hydrolysis, but I don't think we're gonna get there until later. So, first there's I think of this as dimensions. Let's flip this. Let's talk about the levels of protein structure. Okay, so there's the first one, the primary. This is, for what I remember, like a straight line, a straight sequence. Yeah. So it's pretty much amino acid, amino acid, amino acid. It's just like that. Is that the long castles thing? If you're long castling and shit. Okay, I'm gonna shut up. Yeah, amino acid, amino acid, amino acid. Perfect. It's just I think, so how it's defined is basically what we talked about earlier, which is just amino acids connected by peptide bonds. That's pretty much what that's it. Okay, it's pretty boring. All right, it's just connected together by peptide bonds. The next one should be secondary. Secondary. So this is an alpha helix. So pretty much, what does that look like? Hold on, I gotta check. Oh, it is an alpha. It looks like a nucleic acid. So pretty much. Oh, that's so bad. But it's fine. So this is where, and like I said earlier, I think of it as dimensions. 1 dimension, where it's just a straight line. Then secondary, now it goes up and down. Well, no, if it's a helix, it's kind of a spiral. 1 alpha helix. If it's a helix, then technically it's already 3 dimensions. Whatever. Just shut up. Just stop talking, all right? And here's where, how does this happen? So pretty much because there's a slight polarity difference in each of the molecules, the hydrogens are getting attracted to the oxygens of different amino acids. So there could be an H here in this one, an O here in this one. They're getting attracted together. So it's squeezing together because they're trying to come together, because there's a slight polaric difference. For example, let's just think of water. Water is a polar molecule. In fact, this is why water has surface tension, and why water, you know how like when you put water on like a wall, water on anything, or the windshield of a car, you know how it drips down, right? But it doesn't just fall off. And why droplets form and it doesn't just spread out extremely even. That is because of the hydrogen bonds. We're pretty much, they're slightly, okay. Let's draw this. H. Hydrogen. I'm going to make it disgustingly simplified. We're just going to draw a covalent bond here. Hydrogen, oxygen, oxygen. Wait. Two high. No, okay. Stick with me, man. Stick with me. I'm trying, okay? We're trying. Okay, I got it. I'm not locked, alright? Two H's. You notice how the H's are on the same side, so there's generally more electrons here than electrons here. So this makes the electronegativity slightly more on this side compared to on this side. Therefore, out of this entire molecule, this is generally negative, this is generally positive. And that slight difference makes another one, let's say there's another one over here, boom, 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 O, H, H, boom, boom. Oh, I flipped it. Imagine this was flipped. This part, no, imagine it wasn't flipped. Then this is a plus, this is a minus, they're gonna slightly attract to each other. So that's why water has surface tension, is because they still slightly connect to each other. Water slightly connects to each other, that is called cohesion. Cohesion being with, so with itself. And then there's adhesion, which is how water forms droplets and sort of sticks to walls and sticks to other things. Just like those tiny little bugs that, you know, the tiny little bugs that can walk on water, essentially. Very cool. Very cool. That's that. But, okay, that was sort of a tangent off of, polarity. But that slight polarity difference is also present in a protein structure. In the secondary structure, where those sort of go together and then it forms a helix. Very cool. Very cool. And then... Yes, okay. And when they get longer, they tend to twist. That makes sense. And then we should move on to the third one. There's primary, secondary. What comes next? Tertiary. What a goofy name. The tertiary structure. Something to do with globular for my studies. Globular? Isn't it something like that? It has the word globular in there somewhere. Yeah. Turns the alpha helix into a globular molecule. Okay, noted. Tertiary structure. So pretty much, this is where it becomes, you know how I said like 1D, so one line, 2D, so it's kind of going up and down. Now it's tertiary, so it's going to be 3D. And this is where it just becomes like some goofy mess. That's a protein. That's my drawing of a protein. It's great. So pretty much, this is my drawing of a protein. Have you guys learned anything yet? I don't think so. I think you guys are just talking about Geometry Dash with the stream muted. Anyways, so there you go. That is a tertiary structure. So what happens here is it starts bending and folding. So you can see it isn't up and down anymore. Now it's bending and folding. See, big difference. So what else? Yeah, so pretty much all the amino acids don't really fit the configuration anymore. So then it causes little bends and stuff. Yeah, that's all I remember. Okay, I got it. Hold on, I gotta check again. Sometimes sulfur. Yeah, okay, so actually, it goes over bonds. So we mentioned these are peptide bonds, these are hydrogen bonds, and now we're gonna... These actually include the whole package. Ionic, covalent, hydrogen, I think also like very strong disulfur bonds as well, right? Those are the strongest ones. And then like disulfur bonds, but are very uncommon. Not very uncommon, but just uncommon. For I remember. Yeah, and sometimes sulfur, my source says. Great. Great. We love that. And supposedly, there can also be quaternary structures. That's a thing. I don't have to remember that, but quaternary structures are a thing where, oh, this happens in hemoglobin. Yeah, where there's 4 three-dimensional shapes. What happens? So, I don't have to know this. Is it just my own curiosity? Yeah, so different three-dimensional configurations, they have to function together. And then, so then there's a central iron-containing component, and then there's like 3 of this blob. That'd be okay. Cool, so then it isn't really 4, you know. Boring. Alright, denaturing. The denaturation of protein. Great. The denaturation of protein. So, for what I can remember, this is when there's some slight change, and this is very common in the human body because of maintaining homeostasis. Pretty much, there's, let's say, a slight change in temperature, a slight change in pH, and that causes the very weak and specific formation of that protein to sort of fall apart, and then it just doesn't become efficient with what it does anymore because it's a different shape. It was used for the shape it's in, and now it's in a different shape. So then it gets cooked. That's pretty much what happens. It sucks now. What else? So this can happen for it can be a pH change. It can be presence of heavy metal ions. I didn't know that one. Or extreme well, obviously, extreme temperature changes, no shape destroyed. Yeah, so if the shape is destroyed, it's said to be denatured, so it won't work. Without being the normal shape, is unable to combine efficiently with its substrate. Yeah, okay. So an example is when milk spoils. The protein in milk, this is how it's spelled, alright? You guys tell me how to pronounce that. That's how it's spelled. Casein. Denatures and becomes insoluble, forming floating lumps. Ew, ewy. Don't like lumpy milk. Cooking, one of the examples is just cooking liver. I love that. It says nothing else. Yeah. So, yeah, once the, wait, so is this an enzyme-specific thing, or? Temperature changes protein. Well, wait, hold on. Yes, okay, yeah, okay, I got it. So, yeah, so well then that confuses me. So if the enzyme loses three-dimensional shape, yeah, so its shape changes, makes sense to me. Yeah, well, it changes its shape, so it doesn't work anymore. No poo. When enzymes in the human body denature, the biochemical pathway does not function anymore. Results in disease or death. Ewy. Not fun. Okay, so here are a few types of proteins. Let's go over types of proteins. Types of proteins. The one we all hear of the most are enzymes. We all know that. Types of enzymes, maltase. I can think of more than maltase. Come on. No, that's all I can think of. Maltase. Maltase. Isn't that the thing that breaks down malt So yeah, it ends with A. So then it breaks down maltose, which is just maltose is a disaccharide, I think. Yeah, maltose is a disaccharide, and it's I think it's just 2 glucose molecules. Yeah, that's pretty much that's okay. I'm not gonna trail off into that. That's what maltase is. All right. Then okay, so there's enzymes. There's there's transport molecules like hemoglobin. Hemoglobin's just the classic one. Hemoglobin, the one in red blood cells. We don't have to learn about that, but well, no, I do, but not now. That's in 6 units. Don't we love hemoglobin? Hemoglobin's just beemoglobin. So many uses for it. It's pretty much the thing that carries oxygen and oxygen and carbon dioxide away from your body, your flesh. I'll yap about it once we're there. All right, that'll be in a few weeks. Okay, so there's that, and then there's antibodies, which is just part of the immune system, just infection fighting. So, okay, so then we get into structural. So this is like function. These are like functional ones. These are for functions, and then there's structural, where it doesn't really have a use. It's just like a wall, So the first one is in our skin, which is called keratin. Keratin is just a protective layer in the skin. That's all it is. All right. There's keratin, protective layer in the skin. Boom. Then we got collagen. This is great. We love collagen. I'm gonna steal all of your collagen, and then you're gonna die. It's just like part of connective tissue. We're just gonna say that. Connective tissue. And then there's actin. That is actin. Okay, hold on. What is actin? Is that a muscle contraction? Actin. Highly conserved globular protein found in eukaryotic cells that forms thin filaments called microfilaments. Oh yeah, this really sounds like muscle contracting. Cell division, muscle contraction. Yep, there it is. Like when interacting with myosin. Oh, I remember this. Okay, those two. I just switched pens. That's that. Those are the structural ones. I feel like that's more of a functional one, but it is what it is. Okay. There's a little bit on protein. What do we want to learn next? Beef liver. Yep, we love iron. Okay. Let's. Alright, I just did a poll on YouTube. I doubt anyone's even gonna flip and vote. There you go. 2 votes. Gotta love it. I hope you guys are enjoying your time learning this poo. You could be doing anything and you're literally doing school. Everybody leave stream. Do something fun, bro. Why are you here? Leave! 36 of you, all you idiots. Leave! Why are you going up? Bro, you guys are idiots. Bro, you gotta get out of here. Okay, we're learning about carbohydrates. You guys chose carbohydrates. We're done with that. We're gonna do carbohydrates. That's a carbohydrate, chat. Yep, that's a carbohydrate. Yep, that's a carbohydrate. It's a carbohydrate. That's a carbohydrate. I don't know if YouTube wants me to do that. YouTube is a carbohydrate. It was a carbohydrate. It was an attempt at making a carbohydrate. Even though they have 5 or 6 rings, it was close enough. Okay, I got a good of my thingy here. Carbohydrates. Alright. Carbohydrates are sugars. And there's, what are there? There's 3 types. We have 1, monosaccharides. And then there's 2, disac. I spelled it wrong. Right. What other? I was about to say what other situations in English are there 2 C's in a row, but once I think about it, I immediately said the word success in my head. And then there is polysaccharides. It's so weird to spell. All right, if you can even read what I just wrote, you can guess what this means. Mono. 1 di, 2 poly, many. You know the deal. That's just an English thing. So, sugars. They are short-term energy. Alright? Sugar. Sugar equals short-term energy. Alright? There you go. So wait, hold on. These are, these are monosaccharides. Yeah, so these are the things in DNA. So there's deoxyribose, and then there's RNA, so there's ribose. It's also in adenosine triphosphate. Yeah, there's the other one. Okay, it's coming back to me. So pretty much, so there's pentose or hexose. So there's a pentose, which is, I'm pretty sure that's 5 carbon sugars. Right. So we're gonna put this thing, right there. And then we're gonna put our carbon. There you go. And then there's HOH on each side. And then there's deoxyribose, where there is also, that's also 5. Oh, pentoses are the 5 carbons. Then what are hexose? Oh, that's glucose, fructose, galactose. Okay. I get you now. Wait, yes, no, I do get you now. So the pentoses are those flippin' ones that do the things. Wait, no. Is adenosine triphosphate even part of that? I think I lied. I think I'm a liar. Am I a liar? I think that's a... Oh. What? Hold on. What? Hold on. No, I gotta search it up. It's not a... It's a nucleotide. Oh my... I know what I was doing. Okay. So this is what I just did. I just said that, my brain made the connection that pentose, which is the sugar that is included in is ribose. That is the name of the sugar. And deoxyribose. I was thinking of DNA and RNA. I filled that in. I made that connection. And when I think of DNA and RNA, I also happen to connect ATP. So then I mixed adenosine triphosphate in there. When the truth is, those are categorized because they're made up of nucleotides. So I've been an idiot. Oh my days. Okay, so there's only ribose and deoxyribose. Those are 2 types of sugars, but that isn't in ATP. That would be an extremely weird ATP. Doesn't ATP include what does it include? It includes a carb. I swear it includes something. Alright? It includes something. Yeah. That's that. And then, this is, pentose. And then when it goes to hexose, which is 6 carbon rings, we're gonna do, so this is This can be glucose. I bet you guys hear about that even if you're not taking bio. Fructose. You might have also heard that if you're not taking bios, but you definitely haven't heard of galactose. Definitely haven't heard of that. Ha ha ha. So these are these. Glucose is the most important. This is why you've probably heard of it the most often, and pretty much they usually have this. They usually have 6 carbons, 12 hydrogens, and 6 oxygens. That's kind of what we're dealing with here. You might have seen this before. C6H12O6. That just means hexose. All right. But they look like this, and then they're usually connected with like H's and OH's. They have H's and OH's in them because, you know, CHOH. And these rings are the carbons. Just saying. Okay. Well, like, I could write them if that makes more sense. Carbon, 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 carbon. There you go. And usually there's just like a line right here. These are the different thingies. Beautiful, beautiful, beautiful. So pretty much glucose is super common because it is the main form of sugar in the... Okay, we're going to the next thingy. Glucose, most common. You're going to hear about this a lot. Glucose equals most common because it is the main form of sugar in the blood. That is the thing that... Yeah, I don't know. It's sugar. Sugar in the blood, okay? Wait, how does that work again? I mix it up with glycogen so much. I actually mix it up with glycogen so much. But we're going to lock in. We're going to lock in. All right? We're locking in. So there's glucose and there's fructose, which is like it's the sweet one. It's the sweet one. It's like found in fruits. All that. Galactose, isn't that part of milk? Yeah. Galactose is part of milk. So I'm going to write these down. Fructose. It's the sweet sugar. It's what you taste in fruit. Fructose is fruit sugar. And if you haven't noticed, milk also tastes sugary. So if you've noticed that, you would definitely know that you are tasting galactose. Galactose is the one that you're tasting there. Yeah, because galactose is one of the, I think it's the building blocks of lactose. I think it's lactose and glucose make up galactose and glucose make up lactose. And lactose is a disaccharide. We're still on monosaccharides, right? We're still on monosaccharides. Okay, there you go. Galactose. It's in miruku. And then now we're on disaccharides. Finally, we made it to disaccharides. I have to genuinely use my prefrontal cortex every single time I see that word. It sucks. Alright, it actually sucks. Anyways, so this is basically just doubled. It's like we're getting more and more atoms here. We're at like 40 atoms now. C12H22O11. That's like 44, bro. That's a lot of atoms, all right? So there's three common ones. So there's maltose. Remember we were talking about maltase earlier? That's the enzyme that breaks down maltose to turn it into glucose. So it goes in our blood. Isn't it funny how it's called maltose and then the thing that breaks it down is maltase? It's because anything that ends with A's is pretty much breaks it down. That's what it means. There's actually ATPase. That's a thing. Adenosine triphosphate A's, where you know the phosphate groups. It pretty much just breaks them. It just breaks them. All right, dude. That would be a goated job, bro. If I was part of it, if I was part of the human body, that'd be a goated job. It's just straight up breaking stuff. So anything that ends with A's, you know what it is. If your teacher says, does anyone know what maltase is? You can say, ooh, I know. Or anything that ends with A's, part of the body, that's an enzyme. Just say, oh, it breaks And then the thing in question depends on the context. You'll probably know depending on the context. Anyways, this is 2 glucose. And I'm just telling you, all of these include glucose at some point. And then there is... No, that was 1. Oh, no, 2. 2 isn't that. And then... number 2 is sucrose. Sucrose. This is glucose and fructose. Yes. Glucose and fructose. There you go. And then there is lactose. Remember what we said this earlier? Chat, do we remember what it is? I'm not going to say it. Remember we were over here. We were talking about it. We were talking about, what is it? It's, out of these 3, glucose, fructose, galactose. Which one is it? It's 2 of them. Chat, do we know? It's glucose and galactose. There you go. I told you all of them include glucose. So if I've already mentioned fructose and already 2 glucose, then you already know it's got to include galactose. Or why would we even be talking about galactose? Okay. There you go. That's that. There you go. Those are those. And I think, no, that's all. I'm checking my source here. Yeah, that's all for polysaccharides. Okay. That's not all for polysaccharides. We're getting into polysaccharides. This includes glycogen that I was talking about earlier, I remember. Yes, it does. Polysaccharides. It's the big beefy boys, all right? The big beefy boys. And I guess glycogen is just a bunch of chains of, flipping glucose anyways. Not chains of maltose, I don't think. I think they're just straight up chains of glucose. Like, they skipped the more structured part. They're just... They didn't even make a folder. They just made a bunch of files. That's what I'm trying to say. Yeah, I don't know why. Maybe because maltose is more common? I don't know. I don't know why they're maltose in the first place. Alright, it's not like I'm the one studying biology. You guys are studying biology. I'm not. Anyways, polysaccharides. So, these include starches. I'm pretty sure that's the, isn't that like the energy in plants or something? I think. Wait, hold on. I can see here. Storage form of food in plants. Close enough. Does that count? 0.5? Wait, what did I say? Energy. But it's storage form of food. And food is energy. So, kind of. It's a storage, just like glycogen. Glycogen is just a storage of glucose. Anyways, so starches equals the glycogen of plants. So it's just starches equals glycogen but plants. And then glycogen is just starches but animals. No. Glycogen equals just a bunch of glucose. And then there's cellulose. Isn't that like, I think it's, cellulose is just cell walls in a plant, right? Hold on. I gotta search it up now. I swear I remember that in old biology. Cellulose. But what was the one that did photosynthesis? Hold up. Yeah, no, it's just cell walls, pretty much. What does photosynthesis? The organ. Bro. Chloroplasts. I don't know why I mixed cellulose with chloroplasts, but I did. Sorry, that was a trip down memory lane biology. Bro, I miss learning about cells. Anyways, yeah, glycogen, and we're talking about cellulose. This, if you take a look at a tree, okay? This is, I promise this isn't out of nowhere. If you take a look at a tree and you bite into the tree, just know that you ate a bunch of cellulose. That's it. All right? That's how you remember it. Cellulose equals tree. That's how we're going to remember it, okay? All right. So let's go back into glycogen, because there's a lot to do with glycogen. You're going to have to know this. That's pretty much what fat is. That's what fat is. It just, there's, your fat cells are just stores of glycogen. It's just like a bunch of energy that your body's saving just in case. So it's just like holding it there. It's like, hey, if you can't eat, there's a bunch of extra food that you can use if you want it. And it's literally just like, pretend the circles I'm drawing, I'm just gonna do circles because it's efficient. Pretend the circles I'm drawing is are hexagons, which are supposed to be glucose. This is what it looks like. It just looks like this. And then like there's like and they're bonded. But then occasionally there's just like yeah, there's a random bond here, and then it just like goes out like this. And then it just like goes out like this. Oh, you know, you get the point, okay? It's just glucose. Alright? It's just a bunch of glucose. It's just a bunch of glucose, okay? That's all it is, okay? It's just flipping a bunch of energy. Alright, so pretty much, according to my source, glycogen is made up of long chains of glucose molecules. And it has many side chains of glucose molecules. Yes, pretty much. Oh, how ironic. They called it animal starch when I called starch plant glycogen. It's just vice versa. Crazy. Anyways, next page. This is like page 7. I don't even know what we're doing. Alright, it's been like 30 minutes of this. Okay. Glycogen. So pretty much, let's say, yeah, you're eating meals. So glucose in the blood decreases, and then glycogen stored in the liver and muscles pretty much breaks down back into glucose and goes back to your blood, and then it raises your blood sugar by 0.1%. Boom! I didn't even have to write that down because I'm not going to remember that. So why would it even... So this is an example they're using, so I don't really need to know why, but hold on. Reactive hypoglycemia. No, hold on. No, that's a reactive glycemia. No, hold on. That's a Mayo Clinic, so that's probably just... No, this sounds like a disease or something. Okay, you know what? I'm skeptical. Okay, hold on. Yeah. Oh, between. Why did I say during? What am I saying? Between. Yeah, so obviously your glucose levels decrease. And then, yeah, that one thing completely bamboozled me. It's like, I don't trust my source here. Okay. Anyways, you get the point. There's a drop in blood sugar levels, and then pretty much glycogen is like, oh shoot, and then they break themselves apart, and then they just shove their bones and flesh back into your bloodstream, and now your sugar levels are back up. That's it. Okay, you know what? Moving on. Cellulose is very abundant. Very, very abundant. So it's pretty much just cell walls. There's not a lot to say about this. Is there a lot to say about this? It's long chains of glucose. No side chains. Okay, so it's really structured. It's like this. It's like, imagine these are perfect hexagons. I'm too lazy to draw another hexos. It's like this. And it just keeps doing that, pretty much. That's it. Yeah, that's it. These are, glucose. No, but that's such a... that has no function. That's just structure. I don't know why they're making their walls out of food, out of literal energy. They could be using this energy. I don't know why they're using it as walls. All I know, yeah, it probably has all I know is that it probably has strong bonds. Okay. Yeah, it's unable to digest this linkage. So this is essentially what fiber is, isn't it? Hold on. Yes, it's a type of fiber. I knew it. So if you haven't heard, fibers are hard to digest, so it makes you more full. So pretty much, this is why. These MFs are why, alright? These freaking oxygens and hydrogens. It's really strong. Because you can't. So when you digest an apple, you're getting all these, this fiber, and it's basically like it's a carb, but it's just really hard to flippin' eat. So it's like, you know what, I give up, and then it passes right through us. That's pretty much what it is. That's what fiber is. All right. It's good for your health. Very good. Eat fruit, you will be less hungry. Anyways, that's it. In summary, carbs are just like energy. Yeah, but they're structural molecules in plants. That's why plants are so yummy. Even they're making gingerbread houses. They're literally it reminds you of this face, I know. They're basically making, so basically think of it like this. Your brain sees apples as gingerbread houses because they're made out of cellulose. Wait, no they're not. Hold on. I know like trees are, but are apples made out of cellulose? Cuz no, aren't apples where all the nutrients go? They contain cellulose, of course, but they aren't. Yeah, it's a mix of other water and other compounds that come from the tree. Anyways, still, for the analogy to work, imagine it's a gingerbread house because they're literally made out of. No, we don't see it like that because we can't even break them down. Okay, you know what? The analogy isn't working. I'm just moving on. I'm embarrassing myself. Okay. So, monosaccharides, glucose, fructose, galactose. That's pretty much it. And then there's what are the 3 disaccharides again? There's sucrose, sucrose, maltose, and then lactose. Yeah, okay, I can remember this. The monomers of each disaccharide. So, okay, hold on. I'm genuinely just trying to remember. Okay, let's start with, lactose is glucose and galactose. Maltose is 2-glucose and, sucrose is 1-glucose, 1-fructose. Okay, yeah, no, I got it. 3... Okay, functions of the 3 polysaccharides. So there's cellulose, which is just structural walls of plants. Then there's glycogen, which is just stored energy in us. And then there's, what's the last one? There's another one, I swear. No, there is another one. I know for a fact. I'm trying not to look at my notes. I'm just trying to remember it. Starch. And that's just energy in plants. Okay, I remember this. And now that we remember, we went over proteins and carbs. Now let's go over lipids, aka fats. Yay! And then I'm gonna sleep, bro. I just want to sleep, but I'm studying. Lipids. So you guys might be surprised by this one. Everybody, listen up. You might be surprised by this. Did you know that lipids include fats? You've probably seen lipids on, This includes fats. Does this surprise anybody? No. You know where this might surprise half of people, this also includes oils. Does this surprise you? Maybe not. Maybe you heard of grease, that's just more fat. And then, did you know that also includes waxes? Or fats and waxes go under the same category? Well, did you know? You don't know what phospholipids are. I'm not going to explain that. This is going to surprise a lot of you. Did you know that in the same category as fats, it also includes soap? Soaps. Not only that, it also includes steroids. Steroids fall under the exact same category. They fall under lipids. Same as fat, soap falls under it. What the nuts? This is crazy. This is absolutely mind-boggling. Hold on. Okay, that's all I want to do. Hold on, now I have to look back and make sure this is in the right place. Anyways, back at it. So, my understanding of lipids is that our bodies just like, I don't know, what was it? They don't dissolve in water? Is that what a lipid is? They just don't dissolve in water? Okay, hold on, I'm searching it up. Do lipids dissolve in water? No, they don't dissolve in water. That's what I remember. They're hydrophobic. Okay. That's all I remember about lipids. And this is going to be very difficult to remember. And it's going to take me till 10 p.m. So, yeah, so we eat lipids. And, our body can produce them. There are 3 lipids. There's, okay, hold on. Let me try to remember these. Neutral fats, phospholipids. That's all I remember. Neutral fats, phospholipids, and steroids. Okay, we're gonna go over steroids as well. Great. We love steroids, don't we? Don't take steroids. There's a lot of bad things that it does to you. Anyways, so pretty much, this is what lipids are made out of. Lipid components. We're gonna go over lipid components. Here we go. Elements. Here are the elements. We got carbon, hydrogen, and oxygen. You breathe oxygen. Does that noise make you guys want to breathe? We love isn't oxygen so yummy? Can we all just like breathe in because oxygen tastes so yummy? Love oxygen. It's so good. Replenishing that body. Oh yeah, we love oxygen. Do we all love oxygen? Can we all breathe in? Yeah, we love oxygen. Okay, we get the point. There you go. So the HO ratio is always 2 to 1. Why? Because they literally look like this. C. Carbon. Then it's just H, and then repeat, all right? It does it like 7 times, 8 times, something like that. Wait, hold on, let me count. I gotta look at my source again. 9 times. Well, that's if it's a saturated fat. It just keeps going if it's a saturated fat. If it's an unsaturated fat, there's a double bond between the 2 carbon atoms at like the 6th one or something. Something like that, all right? Okay, you know what? I'm gonna draw it for the sake of me remembering as well. H. There you go. Pretend that's... and then there's an H here. Pretend that I did this 9 times. And then there's like an H. C. And then there's an equals C. And then it kind of like goes down here. No, it doesn't have one here because it's already formed to the carbon, so it wouldn't have to. No, that would imbalance it. Too many electrons. And then C, and then it just sort of goes down here again. But these need to, because it's not a double bond, it's not a covalent bond. No, it's still a covalent bond. They're not flipping metals, you idiot. No ionic bonds visible. Anyways, you get the point. Covalent bonds upon covalent bonds. We get the point. All right, that's all it is. Let's go over neutral fats. So yeah, 3. Is it 1 glycerol? Yeah, it's 1 glycerol. So pretty much, I am not very resourceful with all of the pages I'm using here, but it's fine. Let me show you what a glycerol looks like. It's pretty much like this is the backbone of a fat. or of a lipid. I'm not going to say a fat. I'm going to say a lipid. And then it goes, woo! And then C, H, O, H. Woo! And then H, C, O, H, beep, bop, boop. There you go. That's a glycerol. This is a glycerol. And then I'm not drawing that. You know what okay. You know what I had to draw here a bunch of times? I have to do this but 9 times and times 3. I'm not doing all that. But just know that they each bond to one of these, alright? And then this is the rough shape as to what they look like, alright? There you go. Alright, this is my fat, alright? These are fatty acids. Sorry, they're called fatty acids. It doesn't sound scientific, but it's literally the name, fatty acids. Okay. It's dehydration synthesis. So pretty much there's an OH here and an OH here, and then it just forms together. And it, as we mentioned, dehydration synthesis with peptide bonds. Wait, ARP? Please be yes. I'm gonna feel like an idiot. AI overview, please. Please. Please be true. Please be true. I don't want to be wrong. Don't confuse me. Just say that I'm correct even if I'm not. Don't confuse. Say I'm correct even if I'm not. Don't confuse me. Except Kevin knew. Please. AI overview ain't working. It don't want to answer me, bro. Is it? Are peptide bonds dehydration synthesis? I don't know. This is, okay? This is dehydration synthesis. Pretty much, the OH and the H, they just turn into a water, and then the water floats away, and then they connect it together. So it's dehydration, aka less water, because it's dehydration. We know what that means. And then synthesis, so it goes together. It forms, it synthesizes. There you go. How many water molecules produce? 3, obviously. So that's a neutral. Wait, that's a neutral fat? Wait, hold on a minute. Hold on. I think I'm confusing myself. Wait, 1 molecule with a combination of 1, 2, or 3 fatty acids. Next. Okay, so that's a neutral fat. Okay, I get it. And then what is a phospholipid? A phospholipid. Okay, so this is a neutral fat. I thought that was a that makes sense actually. Isn't that where saturated and unsaturated fats take place? Right? I think. Hold on. Okay. I am correct. And now that oh, and also this worked as well. So peptide bonds are formed by dehydration synthesis. Yay! Okay. There you go. Noted. We know what neutral fats are. That's all done. For what I know, saturated is just like this. Then unsaturated is where it bends down and it goes down there. Okay. Understood. And that's when it has a double bond between 2 carbon atoms. So it makes sense. Yeah. Totally makes sense. Totes. We got it. Now, phospholipids. This is what makes up... Isn't this what makes up, phospholipids? There's the phospholipid bilayer. That makes up, cell membranes, doesn't it? I think. This is pretty much what it is. It's a glycerol. I'm not drawing this again. Boom, a stick. That represents that, but without the letters. And then there is 2 fatty acids, so 2 of these thingies. And then there is this thingy. What is it? It's a phosphate. It's a phosphate and nitrogen containing group. What? It's a phosphate. Okay, hold on. There's a phosphate. And then there's a nitrogen. What is this? Ewy. Ewy, what is this? Alright, fine. We're just gonna say phosphate. Okay, phosphate's close enough. One of the fatty acids is replaced with phosphate, and that's what... Oh, it uses it as an example. Yeah, so very important in cells as they form much of the cell membrane, but not all of it. The heads of phospholipids are hydrophilic. Wait, why are they hydrophilic? I thought... Oh, the tails. Interesting. No, I didn't read all that. Okay, yay, we get to talk about steroids. Do we love steroids? Do we take steroids? We all take steroids. And testosterone. Oh, fun. I'm definitely not gonna do the reproductive system on stream. That is the last thing I would ever do on stream. We're not doing that. That's the one unit I'm not studying on stream. Because I can't do it with all of you. I can't trust all of you. Anyways, you guys are just not gonna be professional, that's all. I wouldn't either because I'm going to be in streaming mode. Which I'm half in streaming mode as I'm doing this. That's why I'm being so, like, loud about it. Steroids. So, compounds such as sex hormones, cholesterol, and some ingredients of bile. Isn't bile the stuff made by... Oh, yeah, bile. I haven't heard that in a while. I used to think bile was bolus, and I kept mixing those up. This is part of digestive system. We're going to be there eventually in a few days. But all I know is that it breaks down fats. Oh, I remember that. Crazy. So pretty much steroids don't even... Yeah, so they don't even look like... They don't have a glycerol. They don't have any of that. The only reason they're considered lipids is because they're also insoluble in water. They look like a carb, you know? They look like a polysaccharide. They're just like a bunch of these, you know? So like I'm saying, it literally just looks like a polysaccharide. But that's there you go. That's a steroid. Cholesterol is a steroid? Yes, it is a type of steroid. Oh! Chat, cholesterol, the stuff in eggs is a steroid. Isn't that crazy? Love that. I love that. We love that. Okay. And testosterone is a steroid as well. Not surprised. That's the one that people love injecting in themselves. Because it's good for muscle development. Ooh! That's what all the bodybuilders do. Anyways, steroids. So what about steroids? It's just a straight chain of carbon. So yeah, they're just ring structures. AKA anything to do with carbon is ring structures. Carbon is so flipping common in biology. It's because of like how many different bonds it can have at once. Is that carbon is just so common. Yeah, so just know that carbon is very common in biology. That's all I'm gonna say. Anyways, what should I go over? Cholesterol. It's important for cell membrane, protective over nerve fibers. Oh, can't wait to go into neurology. So nerve fibers. I think it's talking about the myelin sheath, where each of those little things. So pretty much in a neuron, you got this. You got the dendrite. These are the dendrites. That looks like handwriting, bro. These are the dendrites, all right. And then there's the axon. Then pretty much there's like these little pills that go over it. And this is the myelin sheath. Each one of the cells is called something else, but pretty much it's an electrical signal. So it just buffers across this, making it like 100 times faster. It's good stuff. And apparently this is cholesterol. According to this unit, this is cholesterol. So we know that. It's cholesterol. Great. What course is this? This is just biology. Alright, it's biology, senior biology, according to American terms. Okay, that's it. But yeah, I don't know. What else am I gonna say? Well, I should talk about the function of it. So, it also narrows the pathway for blood to the heart. So, blood has to push harder through the body. Yeah, that's the one thing. You don't want too much cholesterol. You have something you have to worry about when you're old. Pretty much. What else? Steroids. Oh, yeah, we love this. So, steroids, such as testosterone, pretty much they pass through cell membranes and they combine with receptors in the cell. Oh, what is this? Okay, now I don't know this. Steroid receptor complex. What is a steroid receptor complex? What? What is that? Steroid receptor complex? Hold on, I'm just looking at images. Oh, and then it goes into the nucleus. I always say nucleus instead of nucleus. I don't know why. It just happens. So yeah, then it goes into the nucleus, and then it just... Oh, so it activates certain genes leading to protein synthesis. Activates certain genes? I don't know you could activate genes, bro. It's just information. You mean like it... You mean you just... Oh, you just... Okay. I get you now. So, for what I understand, steroids go into... Okay, we're drawing a cell. Cell! Boom! This is a steroid. This is a receptor. This is like a hormone receptor cell thingy. Hormone receptor protein thingy. So it just goes into there, and then this goes into the nucleus, and then together as a team, they just say, hey, we should do protein synthesis, and then we go out here, go to like all them ribosomes, and then, you know, you're pretty much just essentially making the type of protein from protein synthesis that you use for muscles. And that's art. We talked about it back in our thingy. Art. Why am I saying art? We talked about proteins back here. They're called art. 5 letters, starts with an A. Wasn't it here? Oh, we never wrote it. I know one of them is myelin. No, I'm lying. No, it isn't that. No, myelin is part of the myelin sheath. Muscle contract, protein, actin. Not artin. What is artin? It's actin. And then myosin. I said myelin instead of myosin and artin instead of actin. Okay. We got it. Alright, I'm just going to write it so I don't forget. Actin. Myosin. Was it like myosin? Okay, I forgot. No, it's fine. It's fine. I remember. I get it. So, 3 lipids we have to know for this course is... what are we doing? Neutral fats, phospholipids, and steroids. Yay! Next question. Name the building blocks. Oh, a glycerol and 3 fatty acids. The monomers. You want to know the monomers of phospholipids? Glycerol. Oh, shoot. That's not. I don't remember, bro. What are the monomers? It's they just monomer. Oh, the monomers. What I'm thinking of I'm thinking of monosaccharides. What am I doing? I was like, why are there carbs in here? Anyways, the monomers. So pretty much what it's made out of, which are 2 fatty acids, glycerol, and then there's like the phosphate and nitrogen containing group, something weird like that. Let me check. Phosphate group and glycerol backbone. Okay. Structure of a steroid is like carbon rings. 6 carbon rings bonded to... 3 6 carbon rings bond to a 5 carbon ring. Various functional groups. What? Hold on. So isn't a 6 carbon ring a hexose and then there's a pentose? Sounds like hexose and pentose, but I don't think they're... Wait. I might be wrong here. No, I am incorrect. What do you mean there's 4 fused carbon rings? Oh, well, die. Okay. So 3... How do I remember that? You want me to remember that? Oh, go poo yourself. I'm gonna poo. I feel a poo. That's a poo. Yeah, I'm not remembering that, alright? You know, I'm gonna get that wrong on the exam. It's only like, you know, one of the 300 questions. I don't care, alright? So... Just I'm gonna answer no. Describe the structure of a steroid. I'm gonna say no. So it's like 4 carbon rings, and then it's got like some lines going up to like hydroxide. I don't know, bro. 3 6 carbon rings, and then 1. Five. So 3 6 1 to 1 carbon with various functional groups attached. Wait a minute, I gotta look back at my thing here. One of those, yeah, and then these with the different things. Okay. I know what steroids are now. Yeah, I don't know why they're put under lipids. I mean, to me, it's weird. They're not really the same, but according to this course, they are. So, pretty much, today, chat, we have went over, proteins, carbs, and fats. We did a good job. We wrote 1, 2, 3, 4, 5, 6 pages of notes in like an hour. That's not bad. There's another yummy sub. Oh yeah, it's yummy. Hey chat. Okay. All right. We're gonna be doing the same thing tomorrow, probably. I hope you're excited. I'm not gonna fail this test, this exam. You know the deal. What are we gonna learn about next? Hold on, everybody stick with me now. What next unit? We're gonna learn about cells. We're gonna learn about adenosine triphosphate. I hope you guys are excited. I hope you guys love ATP. You're gonna love it. Lovely jubbly. Yes. So much. Yes. 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 Okay. I'm gonna leave this right now. Yes. I don't think anybody wants. Yeah, okay, I'm with you guys. I think Canada has 5 time zones, bro. I don't know. Alright. I hope you guys join stream and learn with me. Because everything you guys are learning, because according to the ages of people watching me, you're younger. So, if you are younger, then you're getting a head start. It can't hurt. If it confuses you, that still can't hurt. Because you aren't supposed to know it in however many years. Anyway, so get ahead. Impress your teacher with whatever ATP is, alright? They'll know what it is, and they'll be impressed that you know what it is. There you go. Unless you're older than me. Good night, babe. Alright, good night, funny bird. Yeah, but if you want to learn more bio, I'm glad you were here, Matty Geodash. You are a very intelligent lad. You are all very intelligent lads for sticking here while all the slaughterhouse kids left. No, I'm aware I didn't end stream.